able to identify, comparing using a human patient data set, that these cell line and apoptosis related pathways can predict with a very high statistical significance the chances of a person getting metastasis. And remember, this is at the time of surgery. When a patient gets surgery, if you have the tissue and then you look for these genes, and if you see that they are upregulated, then the clinician can immediately take a decision, no, this is a very aggressive form of cancer, and we have to treat this person very aggressively. So this is working fine. The another pathway which we are very much focusing on, as I said, is the DNA repair pathway. So here again, the same story. Green indicates upregulation of the gene. Green, yeah, in this case, green is upregulation and red is downregulation. Combined, when you take all these genes together, and again go back and look at the, go here first and then I'll come back to that. When we compare that with the human data set, yes, the DNA repair pathway in itself is also predicting with a very high level of statistical significance, chances of metastasis. So what does this tell you? What this tells you is that if anybody has access to these studies, the time when the surgery is done, we will know a lot about the tumor. We will know how the tumor is going to progress. Is the patient going to get metastasis in 5 years, or there is a chance of the patient surviving metastasis free for 20 years? So, someday this would become, like we have the splice variant study going on to phase 1 trial right now, someday this would go to the clinical treatment as well. Now, one thing I want to discuss with you is the DNA repair pathway is also transiently upregulated. What I mean by transiently upregulated? Remember my 4 populations: the average primary tumor cell, the invasive tumor cells, circulating tumor cells, and the lung metastasis. This DNA repair pathway is, with the exception of this gene securin, the others are upregulated only in the invasive cells and circulating tumor cells. So if we can target something to this DNA repair pathway, we will be able to selectively target the invasive and the circulating tumor cells. But we know the DNA repair pathway is needed pretty much in the entire body. So I can't just go and kill the tumor and the patient. So we have to give a drug that will only kill the tumor and not the patient. So designing a drug totally against DNA repair pathway, it's not going to work. It's going to block all the DNA repair in the body, and it's going to be a chaos. So what we started doing, hold on to this thought, because there are 2 more slides which I need to describe before I'll go on to the DNA repair pathway. So remember I told you the biomarker study? One of the biomarker studies that has gone into clinical trial is a biomarker called MENA. Okay? MENA comes in multiple flavors. MENA is a protein that comes with a splice variant called IMV and a splice variant called 11A. A ratio of those can very nicely indicate whether the cancer is going to be aggressive or not. How do we know that? Because we have gone on to clinic. We have taken breast cancer fine needle aspirates and we have looked at the expression of these different splice variants. In fact, Farzana in my lab is doing real-time PCR looking at exactly the DNA repair pathway and the MENA splice variants right now. When she will have data, she'll be able to show you, those of you who read her report, that this is working very nicely. This is working beautifully. We are able to correlate, and I'm not going to show you that data here because it's not yet ready. That the most aggressive cancers have very low amount of this MENA11A and a very high amount of this MENA triple plus or MENA8. So this is the one that Albert Einstein College of Medicine has patented, and we are moving towards clinical trials very soon. So, I spoke about the biomarkers. Let's talk about the pathway-directed drug. I'm just going to show you one slide with one new pathway-directed drug designed by a company called OSI Pharmaceuticals, which is, along with Genentech, is very effective in these invasive and circulating tumor cells. We have tested that. And that is a drug called Tarceva. Tarceva is an epidermal growth factor receptor blocking drug. Normally, Tarceva is not given to a breast cancer patient. It is given to a lung cancer patient. But we have seen that when we treat the cells with Tarceva. Remember when I showed you my last slides? These bars were higher, these two bars were higher, and these two bars were lower. Look at this one. All the bars are nearly the same, indicating that Tarceva kills all the 4 populations equally. Right? So this is a finding. We find that if you give Tarceva to this patient, I don't know about that for the patients right now because there's a long amount of work that's needed before we can conclude that. But at least there is an indication that pathway-directed drug will kill these invasive and circulating tumor cells. Now, let's come to the last part of my talk. Radio and chemosensitizers that can revert these cells back to radio and chemosensitive. Again, complex story, but I had to show this to make a point. You know, as cells replicate, they go from two stages of growth, G1 and G2, and there's a synthetic phase. When you study metastasis, when you study mitosis, when you study meiosis, you will study these in detail. But one of the components of this process, this entire cell cycle process, is a protein called protein phosphatase 2A. Complex name, don't get into it. But basically, when that gets upregulated, there are 2 things happen. The cell cycle is blocked, okay? And cellular structures get reorganized. These 2 big things happen when PP2A is upregulated. In our microarray studies that I told you right at the beginning, we had discovered that this PP2A is upregulated about 3-fold in the invasive cells. So we decided what happens if we block this. Remember, we were searching for pathway-directed drugs which would kill the invasive and the circulating tumor cells. That was the whole aim of my project. So now, once we block this PP2A using a drug called LB1, now this LB1 happens to be a brand new drug. This is discovered by National Institute of Health in Maryland, Bethesda, scientists from there, patented by them, licensed to a company in Long Island, and I'm collaborating with them to work in breast cancer research. They're working in glioblastoma and others. So what I'm trying to tell you people is that there's a lot of new drugs which are at different stages, but a lot of research is still needed to conclude that yes, they will be effective. So what are we doing with this? I just want to remind you that when we gave radiation alone, these 2 populations did not die. When we gave chemotherapy alone, these 2 populations did not die. However, if we give this drug, LB1, along with radiation or along with chemotherapy, we are able to kill both these populations. Isn't that exciting? Yes, now we are able to target the population which would not normally die with chemotherapy. Okay, but this isn't a petri dish. Is this going to hold true in patients? We'll see. But what do we do in the intermediate? In the intermediate, we try animal models. And I think I'll skip over this one. Let's go to animal model. Okay, this is an animal model which we have in our lab where we inject cancer cells and form a tumor. And you can see that a pre-treatment mouse has a huge tumor. Okay? And after 3 rounds of doxorubicin and LB1, the tumor is totally vanished. Looking exciting, right? Okay. These are charts of 2 different models, one a rat model and a mouse model. And in both cases where we give LB1 plus dox, the tumor pretty much regresses. But remember, I told you a few minutes back that we rarely will ever get a patient who has a tumor so small that we will be able to treat it this way. Majority of the time, the patient will come to the clinic with a lump. Tumor is already grown. Now what do you do? So you have to do surgery. You have to remove it. So we started doing a model which is more realistic to the human scenario. Just bear with me, this is my second last slide. We inject about 500,000 cells, form a tumor, 6 weeks, surgically remove the tumor, like it would be done in a patient. Surgically remove the tumor, stitch it, and then give 5 cycles of chemotherapy. When we're giving cycles of chemotherapy, we divide them into 4 groups. LB1 alone, LB1 plus dox, This is the injection regimen that is described here. The groups are actually described here. Control, LB1, dox, LB1 plus dox. Okay? And we give 5 cycles of chemotherapy. At the end of the chemotherapy, we remove the lung and the bone marrow and look at how much cancer has spread. And look at the dramatic result that we have got recently. We can see that in the population that has LB1 plus dox, metastasis is practically reduced. We are very excited about this. We're gonna take this to different levels, and in fact, Frida is working in this project, trying to understand how to estimate the mortality and morbidity of the treated animals, because it is not only important to live, but it is important to live well. So, if any treatment makes you live, but makes your life very miserable, we don't like that either. So we are trying to understand how if this treatment is also going to reduce the stress on the animals. I think I will stop at this with a conclusion that there is a dominant population which is the root of many problems. We are trying to target that. All right? So we have hope. Hope is we have biomarkers that offer earlier detections that can predict and differentiate between bad and worse cancers. We have pathway-directed drugs that kill dormant cells. Radio and chemosensitizers that revert these cells back to radio and chemosensitive. Chemo and radiotherapy becomes effective in resistant cancers. Okay? And the other students have done some part. I have funding from Breast Cancer Alliance, Robert Einstein Cancer Center, I have an industrial contract, National Cancer Institute, and I want to acknowledge all of them. And I particularly want to thank you to go through such a brilliant talk. But please believe me, what we are doing is great. To get exposed to high quality research at an early age is absolutely necessary to make your orientations right, to go into the right professions, and instead of what you could be doing, you could be doing anything during this summer. You have decided to come to work with people like me, and hopefully make a better experience of your summer. All right? Thank you very much, everyone, for listening.